Okay, so we're going to go ahead and look at cell size and how cell size can be manipulated to maximize the exchange of materials between cells or between the cells and their environment. So one of the things that we really look at when we're talking about cell size and stuff is we talk about their surface area to volume ratios. So sometimes we write that like this. And on your equation sheet, you'll have all the surface area and volume equations. So you'll figure out like the surface area for a cylinder, the surface area for a sphere, the surface area for a cube. And so you'll have to identify what type of cell you have. And then all you do is the steps you would take is you're going to calculate the surface area, and then you calculate the volume, and then you divide. The surface area gets divided by the volume, and you get some number. And the higher the number, obviously the larger the surface area to volume ratio is. And that means it's more efficient at exchanging materials in their environment. So it's more efficient at exchanging materials. And so let's look at some examples of specialized ways that has happened. So one of them is in your gut, like in your intestines, you have what are called microvilli, like this, and they're like very wavy like that. And then even if you were to zoom in on those, they would actually like on each of those little, like, let's just zoom in on one bump. So we'll like kind of zoom in on one bump. So here's one bump, and on there's even more. I'm like gonna redraw this section. So if I was like on one bump, there's even more. And so you're maximizing the surface area, maximizing surface area to have the most exchange with the environment. So especially like think about like what is the job of the intestines? To absorb as many nutrients as possible, to have the most exchange with environment. So we see this in a lot of places. We see this. So some examples are root hairs. We see it with, as I said, like the villi in the intestines. We see it in lungs. We're doing all kinds of things to maximize that, where we want to exchange a lot of materials and get as most out of the environment as possible. So that's why cells are doing that. Another example is red blood cells. So ironically, I'm going to get away from the red color when I do this. Red blood cells also are kind of a concave. That's a sickle one. I'm terrible at drawing them, but they kind of, if you were looking down on them, so like if you're kind of like, if I'm looking down, they kind of almost are always depicted like this. And that's because this is like a dip. It's concave. And again, it's trying to maximize surface area. For the volume, so you can get more, so that means that you can get more oxygen can get absorbed. So we're going to do a really fun lab to see this in action and see how the exchange of materials can work that way. Okay.